Alright, we are going to look at the process of translation, which is the process in which the polypeptide is synthesized. Okay, and the synthesis of the polypeptide is with the information stored in the mRNA. Okay, so in the free ribosomes, or the ribosomes bound on the roughly R, if it is a eukaryote, right, the sequence of codons which is found on the mRNA would specify the sequence of amino acids, right, as covered in the genetic code last lesson, right? So the tRNAs, right, would carry the specific amino acids to the ribosomes, and the tRNA would match itself to the codon, right, using the anti-codon sequence, and therefore bring the correct amino acid as specified by the codon to the ribosome to be added to the polypeptide. So this is basically the gist of the process of translation. Alright, so before we look at translation in more details, let's just do a revision on the structure of tRNA, especially the presence of the amino acid binding site at the 3' end and the presence of the anti-codon, right, below, in this orientation, right, below at the bottom of the tRNA, which is a series of 3 nucleotides that's complementary to the codon. Right, so the first step of translation is known as translation preparation, and it is similar in both pro and eukaryotes. All right, and another name to translation preparation is amino acid activation. It is when the amino acid is added to its specific tRNA by the enzyme amino acid tRNA synthetase. Right, so once the amino acid is added to the tRNA, this whole complex is known as the amino acyl tRNA. Okay, so the amino acid is of course added to the amino acid binding site at the 3' end of the tRNA, and this process is specific because the enzyme would be able to recognize the specific amino acid and the specific tRNA, so that a specific amino acid is added only to the specific tRNA. Okay, so in the during the process, right, the amino acid would bind to its specific enzyme, which recognize the amino acid through the R group, right? Because the amino acids, right, only differ in their R groups, right? So obviously the active site should be able to recognize the different shape of the R group. Okay, then after that, the specific tRNA would be bind to the active site. Okay, and again, all the tRNA's molecules are pretty much similar, right? So they only differ in their anti-codon. Okay, so the recognition of the specific tRNA by the active site of the enzyme is also via the anti-codon. Okay, so once that is done, right, then the enzyme would catalyze the addition of the amino acid to the tRNA to form the amino acid tRNA. So now the specific tRNA with the specific anti-codon would carry the specific amino acid to the ribosome. Right, so that would be the next step in translation that we look at in the next video.